Welcome to Vet NeuroJar, the podcast where we delve into the intricate world of veterinary neurology. I am your host, and today we are unpacking a fascinating study that sheds light on recurrent laryngeal neuropathy, or RLN, a significant respiratory disease affecting thoroughbred horses. Recurrent laryngeal neuropathy, as the study highlights, poses a considerable economic burden due to its prevalence in thoroughbreds, with reported rates between 15 and 19%. This condition involves decreased abduction of the left arytenoid cartilage in the larynx, stemming from neurogenic atrophy of the cricoarytenoideus dorsalis muscle. This results in a reduced airway diameter and increased difficulty in breathing. Although the precise cause remains elusive, research suggests it is a peripheral mononeuropathy affecting the recurrent laryngeal nerve. The study emphasizes that genetics play a substantial role in the risk of developing recurrent laryngeal neuropathy, with heritability estimated between 32 and 46%. Prior research has identified suggestive risk areas using single-nucleotide polymorphism genotyping arrays. One such study pinpointed an 8 megabase region on equine chromosome 1 spanning EGR2. Additionally, a genome-wide association study in thoroughbreds revealed a significant link with a single-nucleotide polymorphism upstream of coral, a gene linked to wither height. However, this association disappeared when height was factored into the analysis, indicating that the single-nucleotide polymorphism was a marker for wither height rather than recurrent laryngeal neuropathy itself. This is in line with other studies that found a link between height and recurrent laryngeal neuropathy risk. A critical aspect of investigating disease risk factors for complex conditions like recurrent laryngeal neuropathy is accurate phenotyping. This means that researchers need to be able to clearly differentiate between affected and unaffected horses in order to identify meaningful genetic differences. Historically, recurrent laryngeal neuropathy has been diagnosed through resting and/or exercising upper respiratory tract endoscopic examination. However, the study points out that diagnoses are more consistent when horses are examined during exercise, as opposed to when they are at rest. Several studies indicate that resting endoscopic assessment of laryngeal function can be variable and unreliable in predicting laryngeal function during exercise. Moreover, observer variability is significantly higher when grading arytenoid symmetry during exercise compared to when at rest. This reinforces that exercising upper respiratory tract endoscopic examination is the most accurate and reliable method for clinically diagnosing recurrent laryngeal neuropathy. The study does acknowledge the progressive nature of recurrent laryngeal neuropathy, which makes it difficult to accurately define a control group, especially when evaluating young horses. To address these challenges, the study used ultrasonographic evaluation of the cricoarytenoideus lateralis muscle to refine the control phenotype. This method has been reported to have high sensitivity and specificity in predicting recurrent laryngeal neuropathy. Changes in this muscle may precede functional changes in arytenoid symmetry, making ultrasonography a valuable tool when combined with upper respiratory tract endoscopic examination. Now, let's dive into the methodology used in this study. The researchers performed resting and exercising overground endoscopic examinations on thoroughbreds in a flat race training yard. Horses were included if they were fit and in full race training, and excluded if they had undergone any upper respiratory tract surgical intervention, experienced technical problems, or had differing health and exercise conditions. The researchers recorded each horse's age, sex, height, and pedigree. Before the upper respiratory tract examination, the horses underwent a warm-up period on a mechanical walker. During the upper respiratory tract examination, the researchers placed an endoscope via the right nostril to get a clear view of the larynx, and fitted each horse with a heart rate monitor and global positioning device to record velocity, heart rate, and exercise distance. Jugular venous blood samples were also collected before and after exercise to measure plasma lactate. The horses then walked for 5 to 10 minutes before beginning a low-intensity warm-up, followed by a 900-meter high-intensity gallop up a 2.7-degree incline. The upper respiratory tract video recordings began after the removal of the twitch and stopped when the horse re-entered the yard. All videos were evaluated by an experienced viewer who graded arytenoid asymmetry. Laryngeal ultrasonographic images were also captured and analyzed by an experienced observer who was blinded to the overground endoscopy results. The researchers categorized horses as recurrent laryngeal neuropathy controls if they had equivalent left and right cricoarytenoideus lateralis muscle echogenicity, a resting endoscopic laryngeal grade of 2.1 or less, and an exercising endoscopic laryngeal grade A. Horses were categorized as recurrent laryngeal neuropathy cases if they had an abnormal left cricoarytenoideus lateralis muscle and an exercising endoscopic laryngeal grade B or C. There also had to be complete agreement between the resting and exercising overground endoscopy examination and ultrasonography results for a horse to be categorized as a control, as well as complete agreement between the exercising overground endoscopy examination and ultrasonographic results for a horse to be categorized as a case. To increase the sample size, the researchers also included cases based on a retrospective questionnaire completed by senior managers at the Flat Race Training Yard. The questionnaire was completed for 642 horses born between 2004 and 2015, of which 45 were recorded as cases. The researchers extracted DNA from whole blood and genotyped it using the Illumina Equine SNP70 genotyping bead chip, or the Axiom Equine Genotyping Array. They only included individuals and single-nucleotide polymorphisms with a genotyping rate greater than 95%, and a minor allele frequency threshold greater than 0.05, resulting in a set of 43,831 autosomal single-nucleotide polymorphisms. The researchers then performed principal component analysis, heritability analysis, and a genome-wide association study. They also used HaploView to generate visualizations of the haplotype structure at the equine chromosome 20 locus. Finally, the researchers extracted the 6-index single-nucleotide polymorphisms from archived genotypes for 3,126 thoroughbred horses, including 233 stallions, maintained in a commercial repository. Now let's explore the key findings. The study phenotyped 218 horses for recurrent laryngeal neuropathy using exercising overground endoscopy examinations. After comparing laryngeal ultrasonographic measurements with exercising overground endoscopy results, the researchers found a sensitivity of 75%, a specificity of 93%, and an accuracy of 88%. They therefore used this modality with resting endoscopic results to refine the groups. This resulted in the exclusion of 13 recurrent laryngeal neuropathy cases and 27 controls. In the end, 171 horses were included in the genome-wide association study from the overground endoscopy study. The height data available for the overground endoscopy cohort showed that cases were taller than controls. An additional 64 cases were identified via a questionnaire, resulting in a final genome-wide association study sample set of 235 horses. The principal component analysis revealed no apparent clustering of cases separately from controls. Heritability estimates for recurrent laryngeal neuropathy in the overground endoscopy cohort was 0.41. For the genome-wide association study sample set, heritability for recurrent laryngeal neuropathy was 0.30. The genome-wide association study identified a single-nucleotide polymorphism on equine chromosome 20 that reached genome-wide significance. Applying the clumping method identified 6-index single-nucleotide polymorphisms. The researchers then examined allele and genotype frequencies for the 6-index single-nucleotide polymorphisms. The haplotype structure at the equine chromosome 20 locus was also examined. This defined 4 haplotypes that occurred in 94.1% of cases and 82.2% of controls. The researchers determined that haplotype 2 was a putative risk haplotype, while haplotype 3 was a putative protective haplotype. The frequency of the risk alleles for each index single-nucleotide polymorphism was also examined in a stallion cohort and the general population. What do these results mean? The study emphasizes the challenge of accurately identifying disease risk factors for complex traits and highlights the stringent inclusion/exclusion criteria used for phenotyping recurrent laryngeal neuropathy cases and controls. The control phenotype was defined most stringently, given the progressive nature of recurrent laryngeal neuropathy. While it is possible that some horses in the control group may develop recurrent laryngeal neuropathy as they age, thoroughbreds are at the highest risk of onset between 2 and 3 years of age. The researchers maximized identification of the genetic contribution to the trait by ensuring that the majority of the study horses were bred, managed, and trained at the same premises by one trainer, which controlled for environmental variation. The significant association found on equine chromosome 20 suggests that the genes in this region play a crucial role in recurrent laryngeal neuropathy. Specifically, The top single-nucleotide polymorphism was located between disheveled-associated activator of morphogenesis 2 and leucine-rich repeat and fibronectin type 3 domain containing 2 genes. Given their functions in the nervous system, these 2 genes are strong candidates for recurrent laryngeal neuropathy. Disheveled-associated activator of morphogenesis 2 encodes a formin protein that suppresses oligodendrocyte differentiation and is needed to form the myelin sheath around axons. During development, disheveled-associated activator of morphogenesis 2 is required for myelin compaction, and in adults it is necessary for motor learning. Genetic variants in cytoskeleton-associated genes have been implicated in demyelinating diseases such as Charcot-Marie-Tooth disease. The study notes that Charcot-Marie-Tooth disease is the most similar human pathology to equine recurrent laryngeal neuropathy. Leucine-rich repeat and fibronectin type 3 domain containing 2, also known as synaptic adhesion-like molecule 1, is exclusively expressed in mouse embryonic neurons in the prospective hippocampus and controls synapse development by inducing neurexin clustering in hippocampal neurons. Synaptic adhesion molecules are known to regulate synapse development and function, controlling neural circuit and brain functions. The researchers also identified compelling candidate genes in other regions of interest. The single-nucleotide polymorphism on equine chromosome 1 was located within the adipase phospholipid transporting 10A gene, while the single-nucleotide polymorphism on equine chromosome 14 was less than 400 kilobases from the potassium-calcium activated channel subfamily and member 2 gene. The other equine chromosome 20 region was 20 kilobases from the transcription factor AP2 alpha gene. In addition to identifying candidate genes, another key aim of the study was to identify predictive genetic markers for the risk of developing recurrent laryngeal neuropathy. The researchers investigated whether any previously published markers for recurrent laryngeal neuropathy in horses overlapped with the top 1% of single-nucleotide polymorphisms from their genome-wide association studies. They did not validate any previously published genetic markers for recurrent laryngeal neuropathy other than for height. However, the markers identified in this study appear to be independent of height. The researchers note that examination of the index single-nucleotide polymorphism allele and genotype frequencies, as well as the equine chromosome 20 haplotype occurrence in cases and controls, did not identify a single genetic marker that would provide high predictability for recurrent laryngeal neuropathy in a large proportion of the population. Because of this, the researchers suggest that polygenic risk scores may be a promising approach. They also emphasize the importance of replication or validation analyses in an independent sample to confirm the genome-wide association study findings and the utility of a polygenic risk score for recurrent laryngeal neuropathy risk. The researchers found that almost all the single-nucleotide polymorphism risk alleles segregated in the population, with stallions as carriers. They suggest that judicial breeding using genetic tools could reduce the incidence of risk alleles in the population if stallion and mare genotypes are known. To summarize, this study identified a set of single-nucleotide polymorphisms that may be useful for the development of a polygenic risk score-based screening test to assist in monitoring disease development, which may be beneficial for clinical management of at-risk horses. The researchers also note that integrating these single-nucleotide polymorphisms in breeding decision-making could lend toward the reduction of recurrent laryngeal neuropathy incidence in the population by avoiding risk allele carrier matings. Finally, the identification of candidate genes with functions in nervous system development and neuropathies may also contribute to the understanding of recurrent laryngeal neuropathy pathobiology in thoroughbred horses. That concludes our exploration of recurrent laryngeal neuropathy in thoroughbreds. I hope you enjoyed learning about this complex condition and the cutting-edge research being done to unravel its mysteries. From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.